Hello everyone. Hi, how are you doing? I hope everyone is doing good. So welcome to another video of molecular basis of inheritance. In this video, we are going to learn about what is gene expression and lac operon structure. We have covered up till translation. So this is the last topic of this chapter. Okay. So let's straight away begin with the gene expression. Now, what do we understand by gene expression? If you look at the terminology over here, gene getting expressed. Okay, so this expression is quite regulated. Okay, like you have the, let's say for example, take any gadget or take any device in your house. Let's say take a simple example of a fan. So the fan has a regulator on the switchboard. It has from 1 to 5 number. Okay, what does this regulator does? When you are feeling hot, you regulate the regulator of the fan at 5. You keep it at maximum speed. When you are feeling cold, you might keep it on 1, 2, or 3. Okay, that is, you regulate the speed of the fan by regulating this switch on the switchboard. Similarly, in living system, when we talk about gene expression, this is basically the process of information flow. Okay, how much, what, at what time, in what amount, when the gene should be expressed, when the gene should not be expressed, when the protein is required, not required. So all this is involved in gene expression. So basically, it's the process of information flow from genes to proteins. So whenever let's say a particular protein is required, so that particular gene will be expressed. If that protein is not required, so that gene will not be expressed. Okay, let's say a protein is required in so and so amount, so the gene expression should be there for such a period of time to produce such an amount of protein. Okay, so basically it is controlled at the level of transcription. Okay, we had discussed in detail in the previous videos about transcription and translation. So when the mRNA is being made from the DNA, okay, the DNA gives from the DNA, the mRNA copies the information regarding a particular gene. So at that time, the regulation takes place. That is whether the mRNA is required or not, at in how much amount, till what period of time the mRNA should be there. So all that stuff is there. Okay, so when a protein is required, okay, when an mRNA is made, when transcription happens, then we will call a gene to be turned on. Okay, then we will say, okay, a gene is turned on because that gene which is present on the DNA. Okay, let's say this is a part of the DNA which encodes for a enzyme, yes, which is required in glucose metabolism. So this particular gene is undergoing transcription, means it is make mRNA is getting made from this region of the DNA. Means we will say that this particular region of the DNA, which is the gene for this enzyme, is turned on. It is producing mRNA, which will further translate into a protein. Yes or no? Protein means which will further translate into this enzyme that is required at in any metabolic process at a given time. Okay, so at this stage we will call, okay, the gene is on. Yes, when transcription is taking place. Fine. So organisms will respond to the environmental changes by controlling gene expression. This is what do we understand by expression of the genes. So depending upon the environmental changes, depending upon the metabolic requirements of the cell, okay, the genes will be off and on based on the requirement. Fine. So how do we know this concept? So Francois Jacob and Jacques Monod, these were the two scientists in 1961. They proposed the model of this operon model of gene regulation in bacteria. So the concept of gene expression and its regulation, basically its regulation, was first identified or was first discovered in bacteria. Okay, they were working with the bacteria and then they came up, okay, then they discovered that even these single celled organism, yeah, single-celled organism, prokaryotes, bacteria, even they have very tightly controlled gene regulation. Okay, so one such example that we are going to study is the lac operon model. What is operon? What is it? We are going to see it in a short while. Okay, so the model was based on their study of the gene. So they were working with this bacteria known as E. coli. Okay, so they were working with the genes which codes for breakdown of lactose. Okay, this E. coli bacteria, a particular example we are considering when it makes enzyme to break down the sugar, that is lactose. Okay, to break down the sugar means what? To utilize sugar, to utilize lactose for energy for its growth and multiplication and division. Okay, so they were working with this and then they discovered this operon structure wherein multiple genes are regulated very in a very controlled manner. Fine. So let's begin. So this is how the lactose operon look like. Now, before we move to the structure, we need to know, we are doing the structure. So we have to first understand the different parts of this lactose operon, and then we will understand the working, the regulation of this operon. How does it works? Okay. Now, basically, this word is made up of phrase is made up of two words. Lac stands for lactose. We know what are we talking about? Lactose sugar. What is this sugar? This lactose sugar is used by the E. coli cell, okay, for gaining energy. This energy is utilized by this bacteria to divide, redivide, multiply, expand its population, like that, cell division ke liye. Fine. What is operon? Operon are genes. See, when we talk about a group of genes, if you look at this diagram over here, you can see beta galactosidase gene, permease gene, transacetylase gene. You are aware of this terminology, structural genes, right? When we were doing the transcription and translation, then we saw that mRNA first is has its promoter, then the remaining structural genes, and then the terminator. Okay, so structural genes are the actual genes which are required and copied. The information is copied in the mRNA. So basically, an operon is a cluster of genes. When more than one gene is present, on the same when they are present one after the another, like that, like a big unit. So basically an operon is a regulatory unit. Okay, so we will not only say that it's a cluster of genes, but it is also involved in regulation. Again, what is regulation? We have just discussed regulation. That is, when is this lac operon required? When are the genes of lactose are required? So they will be transcribed. When they are not required, so when this gene has to be on, when this gene has to be off. So this on and off mechanism is the regulation. Okay, so this gene will automatically regulate itself. It will on and it will off depending upon its requirement. Okay, so an operon is basically a regulatory unit. It has few structural genes under the control of one promoter. So there will be only one promoter, okay, and the rest of the genes are under its control. So this promoter will decide whether all the genes has to be transcribed into mRNA or not. Okay, so yes or no, and when do they have to get on, when the transcription has to start, when the transcription has to stop. So all this will be decided by this regulatory region, and these are the genes which will be under the control of this promoter or the operator region. Now we will come to that. Okay, so it encodes polycistronic mRNA. Remember in the previous videos we were discussing about monocistronic mRNA, polycistronic. So mono stands for only one gene. Here we are dealing with more than one gene, right? So that is why poly. Okay, so this lactose operon is coding for genes which is more than one, right? Right? So gene number 1, gene number 2, gene number 3. Three genes are there in this lactose operon. Okay, so that contains the coding sequence for two or more structural genes. So polycystonic mRNA means what? So the mRNA which will be made out of this gene will have three genes. Okay, more than two or more than two. So polycystonic mRNA will be made from lactose operon once transcription begins. So what is this lactose operon at the conclusion? I mean, at the end, what is this lactose operon doing? What is its significance? What is the function of this lactose operon? It is allowing the bacteria to coordinate, regulate a group of genes. These are the group of genes, okay, that has a function which makes protein. What proteins? Enzymes for utilizing lactose. So we are talking about lactose breakdown, which will be lactose is used as energy by the E. coli bacteria for its multiplication and division. So how this lactose molecule is broken down, how this lactose energy is gained from lactose, obviously enzymes will be required. Where what are enzymes? Those are proteins. So enzymes are made. Okay, enzymes are made which are proteins, so protein synthesis has to take place, means transcription translation will take place, okay, from these genes. So they encode the enzymes which are required for breakdown of lactose. Okay, so these are the parts of the lactose operon. Okay, so Uh, basically, in this video, we will just understand the structure. We have understood what is gene expression, what is regulation, that is according to the need of the cell. The gene, any gene, whenever it is required and what amount it is required, when it has to be turned on, when it has to be turned off, everything will be managed, everything will be, taken care of by the cell itself. Here we are talking about the prokaryotic cell in which we have just begun with the example of lactose operon. So in this video, we are just understanding the structure. Okay, and in the next video, we will understand how this lactose operon works. Fine. So up till now, we have covered what is the structural gene. These are the basically three genes which are involved in the metabolism of lactose. So whenever the E. coli is having, let's say you suspend the E. coli in a medium which is having lactose as sugar. Okay, so if you have a solution which is having lactose as a sugar and you introduce 10 cells of E. coli and you want this 10 cells to turn into 1 lakh cells, okay, that is you want the population of the E. coli to multiply, divide and increase. So the sugar will be used as energy. Basically the lactose will be uh, broken down, and the enzymes will be used to break down lactose to gain energy for the cell division and multiplication. Okay, so you have three parts of this regulatory gene, which is lactose operon. So the first part is the promoter. This promoter will start the process of transcription, okay, by binding the enzyme RNA polymerase. What is the process of transcription? That is, an mRNA will be made from this DNA, from this gene. So you require enzyme mRNA, you require enzyme RNA polymerase, which will bind to this promoter over here and will transcribe the genes, that is the beta galactosidase gene, permease gene, transacetylase gene. What are these genes? These genes are required for the breakdown of lactose, that is lactose utilization, through which the energy and nutrients can be taken and the cell E. coli cells can increase, double its population. So this promoter is over here, promoter operator, which will bind the RNA polymerase. Okay, and it will help in the transcription process. Basically, mRNA will be made by this binding of the RNA polymerase. What is this operator doing? Operator region doing over here? Promoter binds RNA polymerase, whereas the operator reach over here, if you will see, This operator region binds the lactose repressor protein. Now, what is this lactose repressor protein? If you see over here, there is one more gene over here which is known as repressor gene. Okay, what is this repressor gene doing? It is making a repressor, lactose repressor protein. Means what? The word is telling you repress, to stop the lactose operon. Okay, to stop the transcription of the lactose operon. So when the gene has to be turned off, when the gene has to be turned off, when transcription does not has to take place, so this operator region will block the RNA polymerase to transcribe further, transcribe these genes which are lying ahead. Okay, so that is why operator is in middle of the promoter and the structural genes. Yes. So this repressor protein will come from this repressor gene. This repressor gene will make a protein, okay, will make a protein. Basically, it will first undergo transcription, translation, and once translation is completed, a protein is ready, okay, which is known as the lactose repressor protein. This protein will be present at the operator region as a lock, when the genes are not required, when the structural genes are not required, so they have to be turned off, they have to be in off position, as in no transcription should take place, so there will be a temporary lock. So that lock will be provided by the operator region, binded to the repressor protein. So this operator region will provide a place, a site for the lactose repressor protein to bind. And the lactose repressor protein will be produced from this repressor gene. It will go and bind to the operator region. It will create a temporary lock or block for this transcription to take place. That is when the gene is in the off position. When it is in the on position, then we will see that scenario also in the next video. So these are the different parts of the lactose operon, and now we have understood their function. Their working, we will understand in the next video. Okay. So this is the LAC-I gene, which makes a repressor protein. Okay, and it will go and bind to the operator region. So basically, this is a separate from the whole operon. It is lies much ahead. It has its own promoter, and then this I promoter makes the lactose protein, and then this lactose protein acts as a tetramer. Okay, and only a small amount of protein is enough for it to bind to the operator region. Okay, so here it will bind. This is the operator binding site. It will bind over here, and here is the lactose binding site. Now that we will see what is this lactose binding site doing in the repressor protein. Okay, about the LAC-I gene, and this is the whole operon, lactose operon. So that's all regarding the structure of the LAC-I gene. Okay, now we move to the structural genes of the lactose operon, beginning with what is LAC-Z, LAC-Y, and LAC-A. Now, what is LAC-Z? These three structural genes, that is, which are required for the lactose metabolism, it encodes for enzyme beta galactosidase. Okay, it encodes for enzyme beta galactosidase. What is this enzyme doing? This enzyme is breaking lactose into glucose and galactose. Lactose is disaccharide made up of two monosaccharides, that is galactose and glucose. These both are monosaccharide. Lactose disaccharide is broken down to monosaccharide. Okay, and it converts the lactose into its isomers, that is galactose, glucose, allolactose. Okay, so LAC-Z is involved in breaking of the lactose molecule. LAC-Y is involved in making an enzyme known as lactose permease, that is giving permission for the cell membrane to allow lactose to come in. Okay, that is membrane protein required for transport of lactose and analogs. So this lactose permease will allow the cell membrane, the plasma membrane of the cell, uh, to allow the entry of the lactose to come inside the cell. Okay, so that is the function of the lactose permease gene, that is the LAC-Y gene. And finally, the transacetylase gene, the LAC-A gene encodes for transacetylase. It basically, once the lactose is inside, it will modify the lactose so that it doesn't move outside the cell. So it is trapped inside the cell. Okay. As you see over here, all the three genes are made one after the another. So you have these three proteins, that is the three enzymes, after transcription and translation is done. Okay, so these are the parts of the lactose operon. So we have understood the promoter region, operator region, and the regulatory region. Then the LAC-I gene, which makes the block, that is the repressor protein, when the genes are not required. We are going to see this in detail in the next video. And finally, the three genes which are encoding for the three important enzymes for breakdown of lactose, or for the utilization of the lactose by the E. coli bacterial cell. This was discovered by Jacob and Monod in E. coli bacterial cells. So Z codes for galactosidase, for Y codes for permease, and A codes for acetylase. So they will undergo transcription translation, and the three enzymes will be synthesized, that is proteins, and then these proteins will be involved in the breakdown or utilization of the lactose. Now in the next video, we are going to see how this operon model works. Okay, when it will be on, when it will be off. So thank you. Take care. Bye bye. See you in the next video.